Hello and welcome back to the HIV RNA TestGuide.com YouTube channel, your trusted source for HIV testing with over 4,500 HIV testing labs across the United States. For more information, check the link in the description of this video or the bio section of our channel.
If you look at the history of global health, the big scientific quests, few challenges have been more persistent, more complex, or frankly more heartbreaking than the search for an effective HIV vaccine. I mean, we're talking more than 40 years. We've seen just immense funding, incredible minds, countless clinical trials that start, they fail, they restart, they evolve. It has just been a scientific marathon, really, one defined by these continuous and deeply frustrating setbacks. And if you're trying to navigate the information landscape today, especially the science news cycle, it is so easy to feel overwhelmed. You see headlines that promise these huge breakthroughs that, they often just don't materialize. But today, we're going to do something different. We are going to deliver a really clear, structured analysis of what looks like genuine progress. We're going to unpack the science that's giving the world real, tangible hope. And the core tension we're exploring is this. The scientific community, for the first time in a long time, really believes we are closer than ever to a viable vaccine.
Oxford University, which is drawing on this massive infrastructure and the success they had with their other large-scale vaccine work, is leading a highly promising next-generation approach.
And that tension you're talking about, that mix of future hope and present-day reality, that is precisely our focus today. For everyone listening right now, our mission here is really twofold. First, we need to spend some real time understanding the science behind Oxford's vaccine candidate, what makes its prime boost and mosaic design so different from all those previous attempts that failed. But second, and this is absolutely critical for actionable knowledge you can use today, we have to understand why, even with this monumental progress on the horizon, the most powerful and immediate tool available to you right now for your own health protection and prevention is still robust early lab testing. And we're going to specifically highlight the gold standard in that early detection, the HIV RNA test. The future is brighter, for sure, but the present demands action, clarity, and control.
Yeah. Exactly. We're going to put all of this into context. We're going to bridge that gap between cutting-edge lab research and, your immediate personal health strategy. So let's lay out the roadmap. We'll start by detailing that decades-long scientific wall that HIV has put up against researchers. Then we'll move to how Oxford managed to leverage existing tech to become a leader. We'll dive deep into the specific, multi-layered prime boost science they're using, and then we'll ground the early results in reality, talk about the timeline, and finally, dedicate a good chunk of time to why that ultra-fast HIV RNA testing is the essential, immediate prevention tool right here in the US and everywhere else. Okay, so let's start at the beginning, the why. Why has HIV vaccine research been such a uniquely brutal, just a taxing challenge for researchers all over the world for 40 years? I think understanding how sophisticated the target is essential to really appreciating how ingenious Oxford's solution might be.
It's so necessary to recognize that HIV is. It's the ultimate biological hurdle. It fundamentally defies the classical rules of vaccinology. I mean, think about it. Traditional vaccines work because they mimic natural immunity. When your body fights off a virus like measles, it eventually clears it, and it develops this persistent protective memory. So the vaccine is just a safe rehearsal of that defense strategy. It's the cheat sheet.
Right. And it gives the body the answers to the test ahead of time.
Exactly. But here's the profound challenge that has crippled vaccine efforts for decades. The scientific research is crystal clear on this. No human being has ever naturally cleared HIV on their own. Not one. The immune system has never given us a template of a successful defense that scientists could copy.
Wow. Okay. That is a staggering realization. Not a single case of natural clearance.
Not one. So traditional vaccine models, which completely rely on mimicking a successful natural immune response, are fundamentally useless against HIV. That fact alone forced researchers into this radical, uncharted territory of what you could call immunological engineering.
So the immune system, for all its power, has never won this fight solo. It's left scientists without a blueprint. But I imagine that's not the only problem. That lack of a blueprint is compounded by the virus's own sort of active defense mechanisms. What are those specific biological roadblocks that make HIV so hard to target, even if you're coming at it with a totally new approach?
There are four primary factors that really form that decades-long scientific wall. The first is just the sheer mind-boggling speed and quantity of mutation. HIV uses an enzyme called reverse transcriptase, and it's notoriously error-prone. It just generates mistakes in its genetic code constantly, every single time it replicates.
So it's making typos on purpose almost.
You could think of it that way. In fact, the mutation rate of HIV is faster than almost any other virus we know of. Within just a single infected individual, the virus can produce millions of variants every single day.
Millions.
Millions. So imagine trying to hit a target that's changing its shape, its color, its location every few minutes. By the time your immune system learns to recognize one version, 1,000 new ones have sprung up, and that renders your antibody defense totally obsolete. It's a phenomenon often called immune escape.
So it's not just evolving over time, it's actively, aggressively generating endless decoys. It's constantly changing its armor to evade being captured. But what if an antibody does manage to identify the target? The virus has a way of physically shielding itself, doesn't it?
Absolutely. And that brings us to the second major roadblock. Its structural defense mechanisms. The outside surface of the HIV virus is covered in this dense, sugary coating called a glycan shield.
Good glycan shield.
Yeah, and this shield acts like camouflage or a physical barrier. It hides the vulnerable, stable regions of the virus that antibodies would ideally need to target. Standard, narrow antibodies just can't penetrate this dense wall of sugar. They literally get deflected before they can reach the conserved parts needed for neutralization.
Okay, so it mutates incredibly fast and has a physical shield. What's the third problem?
The third, and maybe the most insidious difficulty, is its hiding mechanism. It's called latency. See, unlike a lot of viruses that circulate openly in the bloodstream, HIV quickly seeks sanctuary inside the very cells that are designed to fight infection. The body's immune cells, specifically the CD4 plus T cells.
It weaponizes the defense system against itself.
Precisely. And once it's inside, some of the virus can just go dormant. It enters a state called latency, where it integrates its genetic material right into the host cell's DNA.
Oh, wow.
These integrated viral blueprints form what scientists call latent reservoirs. And these reservoirs are completely invisible to the immune system, and crucially, they are undetectable by our current antiretroviral drugs. So the drugs can stop the virus from replicating, but they can't eradicate the dormant virus. This makes a cure almost impossible with current tech, and it demands that a successful vaccine must block the initial infection entirely.
It has to be a preventative shield, not a treatment.
It has to be. And finally, you have the sheer global variety. HIV is categorized into what are called clades A, B, C, D, and so on. Because it replicates and mutates so quickly all over the world, any vaccine has to generate an immune response so broad that it can neutralize not just the common North American subtype B, but potentially dozens or even hundreds of clades at the same time. This need for universal coverage demands a very, very sophisticated, globally aware vaccine design.
Wow. So when you put those four factors together, no natural immunity template to copy, the rapid mutation, that physical glycan shield, and its ability to hide in these latent reservoirs, you really start to understand why this scientific wall was so high. It makes it so clear that since natural immunity wasn't the guide, Oxford had to take a radically different route. They had to essentially engineer a synthetic, universally effective guide from scratch. Okay, so let's unpack the specifics of how they tried to get around these problems.
Well, Oxford's trajectory into becoming a leader in HIV vaccine research is a really fascinating case study. It's all about leveraging infrastructure and prior success. Their achievement with the COVID-19 vaccine wasn't just a medical win, it was an industrial one. It allowed them to validate, to scale, and to optimize a specific technological platform that is now proving instrumental against other complex diseases, including HIV.
So they didn't have to spend years developing the delivery truck, so to speak. They already had it optimized and proven.
That's a perfect analogy. They utilized the same foundational technology that powered their COVID success, viral vector platforms. The platform itself, which often uses a harmless, modified chimpanzee adenovirus called ChAdOx1, is essential because it's a reliable, scalable, and stable delivery system. And in a world of global health, scalability is half the battle.
That is a critical advantage. So their expertise with that specific viral vector, the delivery vehicle, it gave them a huge head start. They had the proven truck, they just needed to fill it with the right, highly complex cargo. The genetic instructions for fighting all these diverse global variants of HIV. But this isn't just an internal Oxford project, is it? We're talking about a global health challenge, which has to require a massive collaborative network.
Oh, absolutely. The collaborative network is essential for success and for validation. The sources all stress this isn't some isolated research in a lab. This work is fueled by massive international organizations like the International AIDS Vaccine Initiative or IAVI. IAVI provides critical clinical trial infrastructure and access to global populations, and they partner directly with academic powerhouses like Scripps Research, which leads a lot of the foundational structural virology and antibody engineering.
I see. And then you have the funding.
Exactly. The robust funding network supported by organizations like the Gates Foundation provide the long-term, high-risk capital that's necessary to pursue these complex multi-year trials. And that collaboration is key because it ensures you have access to diverse populations for clinical testing. That is vital when you're dealing with a virus that has so much global variation across all those clades. If your vaccine only works against subtype B, it's not a global solution.
Right. So if we look specifically at the technology itself, what elevates Oxford's current candidate beyond just having a good delivery truck? What do the sources say are the 4 specific pillars of strength that make their current cargo, the vaccine design, so promising in the face of those 4 biological hurdles we talked about?
So the strategy really breaks down into 4 integrated cutting-edge technologies. The first pillar is powerful computational modeling and AI. Instead of relying purely on expensive and, frankly, slow laboratory experiments to figure out what targets to use, they are employing advanced computing to predict how the virus might mutate, and, crucially, how specific parts of the virus would need to be engineered to elicit a protective response.
That's a huge paradigm shift. Older trials were often based on, educated guesses, or focusing on whatever the most prevalent strain was at the time. This sounds like they're designing the vaccine components with much greater statistical certainty and predictive power.
Precisely. And this modeling directly informs the second pillar, which is the focus on broad neutralizing antibody research and engineering. We call them BNABs for short. The goal here isn't just to produce any antibodies, but to guide the B cells, those are the immune cells responsible for antibody production, to produce these elite, powerful antibodies. These BNABs are engineered to target those conserved, stable regions of the virus that are buried under the glycan shield, effectively bypassing that physical defense mechanism we discussed.
So this is the universal protection aim, an antibody that can neutralize pretty much any major strain of HIV.
That's the goal. The third pillar is advanced T-cell immune system targeting. See, previous vaccines often fail because they focus too heavily on just B cells and making antibodies. Oxford's strategy deliberately and powerfully stimulates the T cells, which are the assassins of the immune system. This dual B cell and T cell targeting is absolutely critical for the unique challenge that HIV presents.
Okay, so you have the predictive design, the focus on elite antibodies, and the powerful T cell activation. What's the 4th pillar that gives it that global solution?
That is the mosaic antigen design. We're going to dive into this more in a bit, but conceptually, this is the strategy for neutralizing that global variety. Instead of training the immune system on a single strain, they expose it to a composite of fragments drawn from countless global strains.
These 4 elements, the predictive design, the elite antibody focus, the powerful T-cell activation, and global coverage. It really sounds like it creates a candidate that is fundamentally more sophisticated and more tailored to HIV's unique evasion tactics than anything that's come before. But does that complexity introduce other challenges, like in manufacturing or regulatory approval? Or maybe it just increases the cost so much that global accessibility becomes a problem right away?
That is an excellent and a very necessary, critical question. The complexity of the design is balanced by the simplicity of the delivery platform. Because they're using that proven, scalable viral vector platform, the ChAdOx1 that powered the COVID vaccine, they've already shown they can manufacture billions of doses relatively cheaply and quickly.
Okay, that makes sense.
The regulatory hurdles for the contents, the mosaic antigens, are certainly higher than for a simple vaccine, but using the established vector technology significantly de-risks the safety profile. The real cost challenge lies in the sheer duration and size of the efficacy trials required. You have to track thousands of participants over many years in diverse locations to prove real-world protection against all of those different clades. That expense is exactly why the Gates Foundation and IAVI funding is so essential.
Okay, let's translate that complexity into action. Now we dive into the mechanism. This is where the innovation really comes alive, where it addresses each of HIV's biological hurdles. We can start with the overall strategy they're using to force this strong, sophisticated response, the prime boost strategy. For everyone listening, you can think of it like the immune system needing 2 highly specialized training sessions, weeks apart. The prime is the initial introduction. It uses the viral vector to deliver the genetic instructions, and it wakes up the B cells and T cells, setting a baseline immune memory. Then, weeks later, the boost is administered. And this boost often uses a different formulation, sometimes a purified protein subunit of the antigen, and it's delivered to dramatically amplify and refine that initial immune response. The objective is to push the immune system to create the broadest and most potent neutralizing antibodies and T-cell armies possible.
That sequential two-part training is absolutely essential. The immune sophistication required to fight HIV just cannot be achieved with a single shot. Can't. It needs that initial blueprint and then a powerful, focused second signal to mature the B cells into producing those high-quality BNABs. So let's deconstruct the 4 scientific components that are loaded into these shots. Number 1, the viral vector technology. We've already established the delivery mechanism uses a harmless modified adenovirus. It's critical to understand why this works. The vector acts like a postal service. It enters the human cell, delivers a package, the genetic instructions to the cell's nucleus, and these instructions tell the cell to temporarily start producing harmless HIV fragments, which are the antigens.
Right.
And crucially, the sources confirm this vector is non-replicating. It can't cause an infection, and it can't integrate into your DNA like HIV does. It is strictly a messenger delivering a blueprint. This ensures safety while providing a potent initial signal to the immune system. It's a textbook learning exercise, not an exposure.
So the vector is safe, but the instructions it delivers have to be specific enough to get past that glycan shield we talked about.
Right.
What makes the actual target so specialized?
That brings us to number 2, broad neutralizing antibody engineering, the BNABs. While older vaccines generated some antibodies, they were too narrow. They only targeted the highly variable parts of the virus. The goal here is different. It's to guide the immune system to produce BNABs that can bind to highly conserved epitopes.
Epitopes.
Those are the stable, unchanging structural areas of the HIV surface protein, the parts that don't mutate. These conserved regions are usually essential for the virus to infect a cell, which means they can't mutate without making the virus non-functional. So the engineered antigens in the vaccine specifically expose these conserved epitopes, forcing the B cells to recognize and attack the virus's Achilles heel. This neutralizes many variants at once, and that's what offers that universal protection aim.
Okay, so that handles the free-floating virus. What about the virus that's already gotten inside a cell?
That leads to number 3, T-cell immune activation. This is maybe the most significant functional difference from the older trials. We have to differentiate between the 2 types of response. Antibodies from B cells, they work against the free virus that's circulating in the blood. But what happens when HIV enters a cell and starts to replicate? You need T cells.
Right, because once the virus is inside the cell, the antibodies can't touch it.
Exactly. The T cells are essential for clearing already infected cells and combating that latent reservoir issue. And there are 2 main T cells at work here. First, you have the CD8 plus cytotoxic T cells, or killer T cells, which recognize and destroy any cell that's displaying HIV fragments on its surface.
The assassins.
The assassins. And second, you have the CD4 plus helper T cells, which are like the conductors of the immune orchestra. They're crucial for coordinating and maximizing the response of both the B cells and the killer T cells. Oxford's strategy ensures a strong, durable activation of both of these T-cell armies, providing a necessary layer of defense against infected cells that antibodies just miss.
That dual approach BNABs for neutralizing the free virus and potent T cells for clearing infected cells, that is the defensive strategy that previous trials really lacked. So how does the vaccine make sure this protection works globally across all those different clades?
That is the role of the final, highly sophisticated piece, the mosaic antigen design. We mentioned HIV is categorized into clades A, B, C, and so on. If you only vaccinate against clade B, you have no protection in vast parts of Africa or Asia where clade C or AE recombinants are dominant.
So it would be a regional vaccine, not a global one.
Exactly. The mosaic antigens are genetically designed sequences that statistically maximize the coverage against the known global diversity of HIV. Instead of choosing a single strain, scientists use computational power to compile a consensus sequence, a carefully curated composite of genetic fragments drawn from a huge range of globally circulating HIV strains.
So it's like creating a police composite sketch for every possible perpetrator worldwide. rather than just showing a picture of one person.
That's a perfect way to put it. This exposes the immune system to a wide array of common features, and it generates an immune response so broad that it can recognize and neutralize HIV no matter what specific clade or strain the person is eventually exposed to. This ability to generate broad, cross-clade protection is the crucial step toward achieving a truly universal HIV vaccine. It leaps over that hurdle of viral variation. So the mechanism is scientifically compelling. It's designed to counter HIV's evasion strategies one by one. But what are the early results actually showing in the clinical trials? The sources confirm the findings from the early trial phases are highly encouraging, but we absolutely have to stick to the rule that early results are not final results.
Right. This is a marathon, not a sprint.
Exactly. Specifically, the early data confirms 3 crucial points. 1, strong T-cell responses, which validates the strategy of activating both those CD4 plus and CD8 plus armies. 2, encouraging antibody activity, which suggests the B&AB engineering is working and the immune system is starting to target those conserved epitopes. And critically, they have demonstrated a good safety profile and a consistent immune response across diverse volunteer populations.
Okay, so interpreting this for the listener. The vaccine is successfully activating the immune system in the precise, predictable, and multi-layered ways that scientists designed it to. That's not just a scientific step, it's a validation of this massive decades-long shift in immunological thinking. The pieces are actually functionally locking into place.
It's a critical functional milestone, yes, because it proves immunogenicity, the ability to generate the desired immune response. But we have to reiterate, this is only the first hurdle. The next phases will have to focus on efficacy.
Efficacy meaning?
Meaning, does this immune response actually translate into preventing infection in a real-world setting? And that is the most difficult and often the most disappointing test in the history of HIV research.
To really appreciate why this consistent immune activation is being celebrated, we have to compare it against the ghosts of past failures, right?
Yeah.
What are the big, historically significant trials that really highlight the paradigm shift that Oxford represents?
When we look back at the historical setbacks, 2 trials really define the difficulty of this task. First, there was the RV144 trial in Thailand back in 2009. This trial generated enormous global optimism. It was the first trial to show any positive signal.
I remember the headlines.
But that signal only resulted in 31% protection. And while that's technically a scientific success, 31% is simply not strong enough to be considered a meaningful public health tool, and the protection it offered waned very quickly.
Okay, so a step forward, but a very small one.
A very small one. Second, there was the follow-up. The HVTN 702 trial in South Africa. This was a multi-year, large-scale effort that was designed to improve upon the RV144 results. Unfortunately, that trial had to be stopped early because the analysis showed no meaningful protection whatsoever.
Wow.
The design just failed to generate antibodies of sufficient quality or strength to overcome the high circulating viral load in that region.
So these failures weren't because of poor science, they're because of the immense biological challenge of HIV. These older models were just too narrow. They targeted fewer strains, they had weak T-cell memory, which is why Oxford's ability to generate this consistent multi-pronged T-cell and BNAB activation using a proven, scalable platform represents a real shift in the paradigm. But let's circle back to that crucial issue of feasibility. We talked about the complexity of the design. Given that the design is so intricate, requiring 2 different administrations, the prime and the boost, and these custom-engineered mosaic antigens, doesn't that suggest the timeline will be even longer? Which would make immediate prevention tools like testing even more vital right now.
That is precisely the connection we need to make. The very complexity that makes it so scientifically promising is what makes the timeline necessarily cautious. While the foundational technology is fast, the regulatory and efficacy hurdles are not. We can't skip the large-scale phase 3 trials.
So what does this all mean for when we might actually see it available for the public? What's the realistic timeline?
The realistic answer is cautious optimism tempered by logistics. Experts are suggesting that the next 3 to 7 years are critical. This extended time window is necessary to conduct those large longitudinal trials across diverse global populations to definitively determine if vaccines like Oxford's can offer strong, durable, real-world protection against acquiring the virus in endemic areas.
So we have a beacon of hope on the scientific horizon. But that horizon is still several years away. This means that today, right now, our focus has to shift from future prevention to present-day prevention. That timeline, that critical 3 to 7 years until efficacy data is proven, means that while the scientific horizon is bright, the immediate reality for you, the listener, remains unchanged. Even with this incredibly hopeful news, testing is not just relevant. It remains the most powerful and proactive prevention and health management tool available today.
This is a point of crucial importance, and we have to make it really clear. A successful vaccine, even a universally effective one, is not going to replace testing. We have to look at this through the lens of public health strategy.
Right.
Even if Oxford's vaccine becomes widely successful, it will not, for instance, eliminate the need for routine surveillance. We still screen for cervical cancer, even with the HPV vaccine. We still screen for hepatitis B, even with that vaccine. A vaccine would simply become a powerful addition to our toolkit, not a replacement for the entire infrastructure that's built around detection, monitoring, and managing existing infections. And of course, the vaccine does not and cannot cure HIV for those already living with it.
And here in the United States, that need for reliable rapid testing is so crucial because we need to maximize that window for intervention. Many infections happen before people even know they're at risk. You get those early non-specific symptoms, often just mistaken for a mild flu, and that can lead to severe long-term health complications if the infection is missed. Early detection is everything. This brings us directly to the modern standard that the research highlights, the HIV RNA test. This is critical information for anyone concerned about their status, and we need to understand exactly what it detects and why it is superior for early screening.
The HIV RNA test is identified as the most powerful diagnostic tool specifically for early detection. It is fundamentally different from the tests most people are familiar with.
Like the rapid finger prick tests.
Exactly. It's different from the antibody-only tests or even the rapid antigen-antibody combination tests. To clarify, an antibody test, the typical rapid finger prick, waits for your body's immune system to react to the virus. And that reaction can take weeks, typically between 3 to 12 weeks. That's the long window period where a person is infected but still tests negative.
Okay, so that's a long time to be in limbo.
A very long time. An antigen-antibody combo test reduces this window slightly. It looks for something called the P24 antigen, which is a structural protein of the virus, alongside the antibodies. This test can usually detect the virus between, say, 2 to 8 weeks after exposure. Better, but still a significant wait.
Right.
Now, the HIV RNA test operates completely differently. It's a molecular test. It does not wait for the body to react. It uses highly sensitive technology called PCR polymerase chain reaction to detect the actual viral genetic material, the RNA that's circulating in the blood. It looks for the virus itself.
So it bypasses the body's entire latency period for generating an immune response.
Yeah.
And that is why the standout statistic from the material is so powerful.
It is the defining feature. The HIV RNA test can detect the virus as early as 7 to 10 days after a potential exposure.
7 to 10 days?
7 to 10 days. That is drastically earlier than any other common HIV test available, and it provides the earliest possible point for actionable health intervention.
That 7 to 10 day window has a massive immediate public health impact. It's the difference between catching the virus when it's just starting to replicate and missing that critical window, which allows the virus to establish those persistent latent reservoirs that we discussed earlier. So let's look at the 3 huge actionable consequences of that early detection that every listener should really internalize. First, early detection means immediate access to early treatment. If someone learns their status in those first critical 7 to 10 days, they can start antiretroviral therapy, or RT, much, much faster. And starting RT during this acute infection phase is scientifically proven to limit the damage the virus does to the immune system right from the start.
Which leads directly to the second point. Early treatment means drastically better long-term health and prognosis. Starting treatment quickly minimizes the initial seeding of those latent reservoirs. Studies show that people who begin RT shortly after infection maintain vastly stronger immune systems, and that leads to near-normal lifespans and excellent long-term health outcomes. That speed is paramount.
And third, and this is maybe the most critical public health consequence globally, early treatment means rapidly reducing transmission.
This is the goal of treatment as prevention. Successful treatment rapidly lowers the viral load to undetectable levels, often within weeks. And we know that undetectable equals untransmittable, UU.
Right.
By catching the infection in those first 10 days, an individual can quickly achieve viral suppression, rendering them incapable of sexually transmitting the virus to a partner. And beyond that, knowing your status early can inform decisions about post-exposure prophylaxis, or PP, for partners. Or it can allow individuals to immediately transition to prevention strategies like pre-PP if they test negative but have ongoing risk.
So the core message here is that reliable modern testing allows you to take immediate proactive control of your health trajectory and protect your partners faster than any other tool out there. That immediate actionable control is something no future vaccine, which is still years away from real-world efficacy data, can currently replace.
Absolutely. As we patiently wait for these monumental scientific breakthroughs, early detection, powered by the sensitivity of the HIV RNA test, remains your strongest and most immediate defense today. So as we start to wrap up this deep dive, let's look ahead. A successful vaccine like Oxford's carries massive systemic implications that go way beyond scientific journals. It affects global health strategy, including right here in the United States.
And what would that look like in practice?
What this means, first and foremost, is more and better prevention options. A vaccine wouldn't get rid of existing tools. It would add a powerful, potentially permanent layer of protection alongside what we already have. Behavioral awareness, condoms, the use of PrEP, and immediate detection via RNA testing. It just gives clinicians and patients more flexibility and greater confidence in prevention efforts.
And structurally, the sources suggest that this would lead, over time, to a huge reduction in new infections.
For sure. And fewer infections mean less operational strain on healthcare systems, less funding being diverted to crisis management, and really importantly, less stigma surrounding HIV. When the spread slows, the perception shifts fundamentally. HIV moves from being this epidemic threat toward being a manageable, preventable chronic condition.
And a global vaccine has global implications.
Exactly. The success of a globally aware vaccine is vital for stronger global HIV efforts and health security. HIV is a global virus. If a vaccine can achieve widespread efficacy, it fundamentally changes the dynamics in low-resource countries, where high viral circulation often drives mutation and the evolution of new strains. Halting outbreaks there ultimately benefits the U.S. indirectly by limiting the importation and spread of new, potentially drug-resistant strains. This is a crucial element of global health security.
And then there's the human element, the hope.
Yes, and perhaps the most inspiring implication is the new hope for younger generations. For 4 decades, HIV has represented this existential threat, a defining medical reality. A successful vaccine creates a future where HIV is preventable, controllable, and potentially truly eliminated from the new infection cycle. It removes the psychological burden that has characterized life for the last 40 years.
It's important to remember, though, that while Oxford is leading this specific viral vector and mosaic antigen approach, they're not the only players pushing the boundaries. The scientific validation of their complex approach has fueled a rush of parallel innovation. The next decade of research is already underway with multiple high-potential pipelines.
That parallel context is so essential. The field is just exploding, leveraging breakthroughs from other areas of vaccinology. Besides the viral vector approach Oxford is using, we're seeing rapid development in mRNA-based vaccines for HIV, which, gained massive traction and validation through the COVID pandemic. It showed proof of concept for rapid deployment.
So that's another whole avenue of research.
It is, and there's also this exciting, highly specialized work on what are called gene-edited antibody vaccines. This research involves using harmless viruses to deliver genetic instructions that program the body's muscle cells to become these permanent, tiny factories that are constantly producing protective, broad neutralizing antibodies.
Wow, so it's not a traditional vaccine, it's more like a long-term therapeutic.
It's a form of passive immunization that could offer long-lasting protection with a single treatment. And finally, we're seeing specialized therapeutic vaccines being developed for people already living with HIV. These aren't for prevention. They're designed to boost the immune system's ability to control or manage the virus without having to rely on daily antiretroviral medication. This synergy between prevention and therapeutic research is just accelerating innovation across the board.
The pace of this research means that the next decade could completely reshape the fight against HIV, leveraging technologies we couldn't even conceptualize 10 years ago. It's an incredibly exciting time to be following the science, but that excitement cannot distract us from the immediate reality.
So wrapping up this deep dive, the future holds immense scientifically validated promise. And that's thanks to the foundational work by Oxford and its global partners. The era of a vaccine is closer than it has ever been before. But until that vaccine crosses the efficacy finish line in the next 3 to 7 years, the smartest, safest, and most powerful approach remains entirely within your hands today. And that approach is simple. Stay informed about prevention options like PrEP, get tested regularly, and if you're concerned about a recent exposure, advocate for and utilize modern molecular tools like the HIV RNA test.
The final synthesis of everything we've covered today is really simple. Knowledge about this sophisticated science, combined with immediate, proactive testing, equals protection. We now know that the scientific breakthrough is real and incredibly sophisticated, but we also know the power of that early detection window, that crucial 7 to 10 days. Oxford's vaccine is a monumental scientific step forward. It's a beacon of hope for a future without new infections, leveraging cutting-edge molecular design. But right now, the most effective step you could take, the quickest, most impactful action, is knowing your status early and with certainty.
Testing gives you control over your health trajectory. Testing gives you clarity regarding your prevention needs. And testing gives you peace of mind, allowing you to move forward confidently. As we patiently wait for these revolutionary vaccines to prove their real-world efficacy, early detection, powered by tools like the HIV RNA test, remains your strongest and most immediate defense.
Thank you for joining us on this deep dive into the science that is reshaping the future of HIV prevention.